distinction is the developmental biology course is really focuses on the cellular and molecular and genetic aspects of development, and not necessarily with an emphasis on human. So along with a prior pre-health professional faculty member, Dr. Patricia Brewer, we decided to put this course together to serve the pre-health professional students at UTSA, or those individuals that are interested in the human embryology. So both she and I trained in departments of anatomy at medical schools, so we took a special human embryology course. Now in medical schools, they integrate it into the human gross anatomy course, and thereby no one really gets a full human embryology course anymore in medical schools. So we were really pleased to be able to offer it, and it's a great course. We don't get to spend as much time on the topic as you would if you were in medical school as a medical student. So it is not as intensive and comprehensive, all-inclusive as it would be there, but it's pretty darn close to it. So it is an upper division course, the 3663, and it'll require you to spend quite a bit of time reading and thinking about it. So my name is Clyde Felix. As I said, I have a PhD in human anatomy, trained at in the basic science department of a medical school. My office is listed here. However, since this course is distance learning, and I've now adopted WebCT for all of my courses, there probably won't be all that much need for office visits, because a lot of what we need to do, and I'll go to WebCT in just a moment to show it to you, a lot of what we might have tried to accomplish through an office visit, we can do through the WebCT. I will be available, if not humanly in person here, virtually through the touch of the button in front of you, or on a phone if you need a more private conference after class, each of our class periods. So we keep the office hours basically posted through this office in the science building, although I won't really be there from 3 to 3:30, but still posted 3 to 3:30 afterward. I always have my mailbox in the biology department office. I'm also the Associate Dean for Academic Affairs, and that's this office number that is posted here. This is my voicemail. I strongly encourage you never to use this. I'm not very good at checking it. This is the email outside of WebCT. I also use that primarily for work-related issues, not for teaching, but you can send me email to that. Just don't expect me to reply, because for this course, we will correspond strictly through WebCT. WebCT has its own mailbox system within it, so when I log in there on a regular basis, if I see that there is new mail, then I know it has to do with the class. If I log on to my UTSA EDU, there's page after page of new emails, and I try to get through them every day as much as I can. And if I see a student sending me something there, there's always, oh, I'll get to that later. And then the next day, it's already a page off the screen, and then the next day, another page off the screen. So please use the WebCT. I'm not saying don't use this, but if you use it with that understanding. Same thing with the 5498. Any communication you have, you can do through the WebCT. As you'll discover, we'll be administering the exams through the WebCT as well. It's extremely flexible, works quite reliably. If there's glitches, we work it out, so there's no real urgency. We can always take care of everything. The WebCT address is shown here, and we'll go to that in just a moment. There are 3 classrooms linked through distance learning on this, downtown. And I guess I should check. You can hear me downtown all right? Someone raise their hand if they can hear me. You see me in that little box? little pip in the side, okay. And at Brooks City Base, Building 559, Classroom 5. We haven't had much in recruiting students to go to the classroom there, but we're continuing to develop our presence on Brooks City Base, so we continue to include it. There might be times where I'll be lecturing from there, lecturing from downtown or from here, so it's kind of convenient for me that way. I can take care of business at those different locations all over the city and still get the lectures produced. Each lecture will be captured live as we're doing it for streaming video. I hope also to capture some of the lectures, as many as I can this semester, so that I have the complete lecture from start to finish, or I may split them so they're a little bit smaller, not the full 50 minutes, and thereby when you download them off campus, if you're not using DSL or the Roadrunner or that type of a system, if you're using the phone line, it'll go a little bit faster, and you won't have to be on the line for a full 50 minutes or 60 minutes, things like that. So all the lecture material will be posted in that way as well. I won't make that announcement right now. I'll hold on that one. There are a couple of prerequisites for this course, although the assumption is that you've had the Bio 1 and Bio 2. This is the previous listing, so if you've had Bio 1 and Bio 2, the new Bio 1 and Bio 2, you'll have even more detail understanding to start with. But you'll catch up quick. We review a lot of things, the essential information, very early on. There's a lot of new information that emerges, so you'll be spending a lot of time really memorizing new terminology, building three-dimensional visualization, and linking it with auditory descriptions. So it's essential that you view the lectures and you try to put together the pictures and the words in your mind. Today our exercise will be a bit of a mixture of words and clicking to a website to take a look at how things look. The rest of the lectures will have either figures scanned from our textbook or from other sources built in, and so it's this combination of the audio descriptions of things and the visual viewing of things, and trying to piece that together so that we can struggle to regenerate in our minds this three-dimensional recreation of a very complex series of events. I don't use any movies in the class. There are tons of movies out there. You can rent them from the libraries. You can see them if they happen to come on television and things like that. Some of the local video stores may even have some of the classics. So I don't bring those in because we've got enough material to cover using our ears and our eyes as it is. The textbook is The Developing Human, Clinically Oriented Embryology, this text here. So if you haven't picked that up yet, be sure that you do, because we jump right into it. We do perform, as I said, an anatomical embryology. Occasionally we'll talk about some of the chemicals involved, some of the signaling, and the impacts that have, but that's not the focus of the course. The focus of the course is that you understand the morphological, anatomical events that are occurring at certain times and get a good feeling for the cytogenesis, development, the cellular development that's occurring, the histogenesis, the generation of tissues, organogenesis, the development of the organs, and organismic development, the overall development of the organism. So that's really the goal, is to have that clear understanding of the distinctions of each of those. So since I know everyone has taken Bio 1, who can tell me what are the levels of structural organization? Hands are shooting up all over. What's the smallest level of structural organization? Chemical, right? And then next, cellular, After cellular, tissue. After tissue is organ. After organ is organ system. And after organ system is organism. So we do touch on all those levels, a little bit on the chemistry. As I said, the developmental biology is really the focus on the chemical level, impact on cellular level, and they do deal with the rest. Our class focuses on the human embryo development. So I do ask that you are courteous in class. You'll notice that there are printers in the classroom. So if you're going to print out, try to do it, please, before class if you're going to be in the classroom. I may be at other locations, and I still see what you're doing on the camera, so I always have the input of what's happening. And the class last year, everybody was printing through the whole class. Nobody complained, but that was fine. Individuals would answer their cell phones and sit there and talk. I didn't scream and holler because I'm being videotaped. But I sure would hope that you would be courteous enough not to do that to your classmates. In addition, you have to realize that your activities on the computers in the rooms are monitored, so you are expected to use them to load the PowerPoint presentation and follow it as we are ongoing, so that you don't have to worry if the signal that is sent is not at the magnification or resolution that you would need to see it. So other activities on the internet are restricted and, as I said, monitored. So behave yourself. If you see an individual at an inappropriate website, you bring it to my attention, and we will follow through the full extent within the university to make sure that individual suffers the consequences. And I say that because it has been reported. So behave yourselves and act like responsible professional people. You're at an academic setting. Okay, not that I'm accusing anyone. It's always hard to say those things and not make people feel a little bit edgy. What does he think I am? Anyway, just because it's happened, I feel I need to mention it. So you see the syllabus is here. Next Monday is Labor Day, so that's our holiday. We push through. Each exam date is listed. You see the census date. Last date to withdraw without a grade is September 10. So it is posted. You have been officially notified. I double-checked it. You double-check it. If I'm wrong, let me know immediately because it's not a good thing for you to be misinformed if that is not correct. I am 99.99% confident that that is correct, so keep an eye on it. Exam 1 is on September 22nd. And you see Exam 2 comes thereafter, so the chapters that you're responsible for and the corresponding lectures are all posted here. So it's really an excellent syllabus. I don't take credit for it. I give credit to Dr. Brewer. As I mentioned before, she had it put together, and she was very kind to provide it to me, and I really appreciate it. So Exam 3 comes up in November, and then we'll have the Thanksgiving break somewhere before then, and the final exam, which is scheduled for the 11th of December, Thursday, 1:30 to 4:15 p.m., and then the summary of the exam schedule is shown here. I'll expect you to read over the part on the scholastic dishonesty. Because the exams will be posted on the WebCT and don't quite yet have a system in place to restrict either the computers that you would use to have access or the location where you would take the exams, I ask that you be honorable persons and limit your resources available to you when you're performing the exam to those that I indicate you are allowed to use. The grade will be determined by the scores on each exam. So here you see the grade that corresponds to the percentage and then the number of points that correspond to that. So the maximum number of points are 500 points that you can get from the 4 exams, and you'll see that as you go along. As you accumulate points, you determine where you stand. Once you get to the grade you want, then you can feel happy about it. That's the grade. I always refer to it as collecting pennies in a bottle. When you get the number of pennies you want in there, then you are confident that they're there. Nothing changes that. So when you achieve, if you get 398 or 400 points, you know you've got a B. Nothing will change that. It can only increase if you get more points. So that's what I like about this approach, and of course, overall, it equals out here as well. So let's now go to the UTSA at EDU, the UTSA.edu. Come on, wake up. Wake up. Here. Have a little picture in a picture on there, so I've got to keep it awake and active so I know it's not blocking what we're lecturing on. So this has changed a bit. I had been easily under control to navigate it. Takes me a little bit longer to get past this one, but I'll get just as accustomed to it as we go along. So when you click on academics, darn, I had it. Where is it? Pretty amazing how the human brain gets conditioned, isn't it? Is it here under the quick links? Webmail. WebCT, there it is. Okay. So on the first screen, with the quick links, is Banner ASAP and WebCT. So we click there and go to WebCT. Anybody in the class used WebCT on another course? Okay. So you're familiar with it. For those of you that it's the first time, you see right here the student login instructions. So you'd click there and follow the instructions to log on. If you have problems, you can contact WebCT. The phone number is shown down here, 458-4519. In addition, you can email WebCT at utsa.edu. I can't really do much for you other than tell you that again if you ask me. So if you need assistance, go straight to them. So once you log on to the WebCT, all the courses that you are registered for that are using WebCT will be posted there for you when you get to the My WebCT home. So you can see that even my summer courses are still listed there. There's new mail. I guess I should check that sometime. But that's how you will know if there's any emails that I have sent to you. That prompting right down here, see, human embryology, will be there. It'll say there are new mail messages. You can communicate with each other and leave me out of it. So you can communicate when you do a message on there. I'll show you in just a second. You can select who you're going to send it to. So when you click on our course, the human embryology, it'll pop up immediately. As long as you don't have high expectations for immediately all the time. So you see there's the course content, calendar of events, grades, the syllabus, and PowerPoint lectures. Streaming video will be added to this. As I said, each class will be posted. I probably won't have the chance to be very good at the calendar of events. There's one other thing that I need to add on here. So let me just add it quickly since we want it on there. Communication, mail. Put it on the navigation bar on the organizer page, the default icon. Okay, so now we now have mailbox on there as well. And when you come to the mailbox, you can compose a mail message, and then when you go to the browse here, it lists everyone on there. I should be on there too. I guess they haven't upgraded it yet or something, or updated it. I think there's more people than that, and usually my name is on there too. Student Felix will come to me? Okay. But you can pick, one other individual or the whole group to communicate. You, of course, are able to attach files. So if there's any type of thing you need to get to me that would be a file attachment, we don't have any such assignments in class, but you can attach a file if you need to. So this is the way that we will communicate. If I send you an email when you log in, it'll say there's new mail. If you send me something, it'll say there's new mail. And you're free to get upset with me if I don't check it soon enough. And let me know that you're upset about it. And that'll be my spanking. I do everything I can to be good at it. So the syllabus that we just covered is posted here. So you can print it here in the room that you're sitting now to have that with you when you leave today, and it's always available here. So as well, all the PowerPoint presentations are there listed by lecture. So we don't have them posted by date or by title. You can call these up. Let's see how it's going to do here. You can right-click, go to the full screen. I hit escape, and it went back like that, the ESC button on the keyboard. You can right-click and print. When you print, you can do the setup. Where is that? Down here, is that right? Also, if you're going to do the handouts, then you can select how many per page. If you do the 3, or I guess the 3 per page, then you get the lines on the side for, oh, I'm not paying attention to my pip there. You get the lines on the side to take notes in class. So that will help you to take care of that component of the class. In addition, the course content for each lecture. And again, I acknowledge Dr. Brewer for preparing this. I prepared the PowerPoint. She prepared all of these. So you have the outlines. These outlines for each of the lectures I used to create the PowerPoint presentations. So everything that you need to know for the class is within these, and then these are within the PowerPoint along with figures from our text and other figures that supplement the materials from our class textbook. So I'm going to exit that, and if you all want to, we're going to go to Lecture 1 to start the lecturing now, but there is a website, this one that's shown here, that I'll be clicking back and forth onto as we now begin mostly the verbal discussion of the milestones in development, and then come here to take a quick look at a picture. So this website is what I'll do so it can be more easily read. I'll go to the PowerPoint, Control Home. No. I'll insert a new slide. I'll paste it here, and we'll start the show. So this is the web page that will be supplement to Lecture 1. If you want to enter that in, get that loaded in your Internet Explorer, in addition to having Lecture 1 posted from WebCT. If you don't have access to WebCT yet, it's all right. Mostly what we're going to do today is just kind of cruise through some of the major milestones, and it is primarily reading, and we'll pick up where we leave off at the lecture on Wednesday. So that's www.visembryo.com/baby/hp.html. Let me get my pointer set on arrow. So there's a tremendous amount of terminology that will be presented in this class, but there are also very key concepts that we need to establish. One is for curiosity, because we all develop this way. We all are involved with families being generated and created, or having our own babies ourselves and so forth. So there's that interest. But then in addition, it's expected that, or not an expectation, but typically most of the people that take this class are biology majors. If you get a degree in biology, then you basically have the obligation in the general public to be able to inform people when they inquire about things. And with stem cell research and abortion and all the key issues, in vitro fertilization, cloning, when all these topics are of really key concern in society, then you have the obligation of sharing your knowledge to help clarify things with people. So a good, strong background in this area is essential to meeting that commitment. So there are two major developmental periods: the prenatal, figures 1, 1, 1, 2 from the text, which is fertilization to birth. And then there is the postnatal, and that's birth to development. So developmental biology really is the study from conception to death. It's the complete spectrum of development, from the moment we're conceived to the moment that the life force leaves the living organism. Embryology, of course, really is more limited in its studies, and it's the prenatal portion that we are going to investigate. So the human reproduction and development really has a lot to do with the evolutionary success of humans, and in part that's related to the internal fertilization and the uterine or intrauterine development of the embryo. There is the placental trait. So are humans viviparous or oviparous? What about the shark? The kids and I would go through this every time we'd go through the shark thing at SeaWorld. They are oviviparous. They hatch eggs, but the eggs hatch inside, and then they give birth to live animals. So humans are viviparous, and there is intrauterine development, of course, thereby. But we do have a unique kind of placenta as well that has a hemochorion. There is the open blood lakes in the human uterus, the endometrium, with the implantation of the embryo. In some species there's placenta, but not the hemochorion. There isn't the opening of the vasculature. So these are some of the key things that we keep in mind about commonalities among mammalian species and uniquenesses of the human situation. The postnatal nutrition of the newborn is via the maternal lactation, a mammalian trait, of course. The development of the limbic brain is key for the nurturing because are young are not precocious. They require a tremendous amount of nurturing. And then the development of homeostatic mechanisms, the avian and mammalian traits that are shared. So there's a significant relationship between embryology and the evolutionarily successful human species. The human development then follows this line of events. There is gametogenesis, the formation of the haploid gametes, the sperm and the eggs. There is fertilization with the fusion of the sperm and the egg to form the zygote. So there is a term for the outcome of conception up until a very key point, and it is the zygote. So we will use specific terminology. We won't use the term embryo until we reach a certain developmental stage that morphologically satisfies the criteria in the definition of embryo. As I pointed out earlier, I asked Jennifer if she's 8 weeks. If she isn't 8 weeks, then we don't refer to the conceptus inside her as the fetus. It is still the embryo. So there's very strict definition of these stages of development. So you may find individuals referring to the embryo erroneously in textbooks or in the literature based upon that strict definition that we'll be using. Cleavage is a special rapid mitotic cell division of the zygote to form the multicellular embryo. So we'll see that there's some unique things about these early cellular developments that we don't always see in other cases of mitotically active cells. And specifically, that is that you'll have cellular division without production of new cytoplasm. So the egg is a very large cell, and it will be split up into equal portions, basically, to produce the first small number of cells. I've forgotten exactly when it is. It's the 8 or 16 cell stage, something like that, before new cytoplasm is going to be developed. So this minimizes the metabolic needs of the cell very early on, and anatomically at that point, of course, we don't have implantation, so the source of nutrients is just the solution that is within the fallopian tube. So these are key things to keep in mind. What limits the development, the rate of development and growth of the conceptus? So when I use this term conceptus, even though we didn't get to all the definitions yet, I'm referring to any stage along the way from fertilization to birth. Whatever there is, the collective group of cells, which is, of course, the chorion, placenta, and all the membranes, and the developing organism, whether it's zygote, embryo, or fetus. So that collective embodiment of cells is the conceptus, everything that was produced by conception. So I use that term when I don't want to be differentiating any particular specific component or part of the developing cells. So another aspect of development is morphogenetic movements. So here we're not talking about genetic like the gene. The term gene originated from genesis, to produce. So here we're talking about the genesis or the production of morphology. So there's cytogenesis is a term that we've used already, the development of the cells. Histogenesis, the development of the tissue. Organogenesis, and development of the organism. So organismogenesis, we stopped using it at about that point. So the development of the morphological, the characteristic morphological appearances occurs really through specialized types of movements. It'll be sheets of cells or clusters of cells that are migrating, or individual cells that are migrating. So there'll be a variety of things that we're going to have to try to envision that are occurring to produce the characteristic feature, three-dimensional structure that we see. In addition, we'll be looking at two-dimensional structures, just sections through the developing conceptus to try to make sense of what's happening. So there's cell and tissue differentiation. So the part of cytogenesis is differentiation. We will talk about mitosis and meiosis, and one of the first things that I would do when I was teaching the BIO1, and I still bring it up when I teach the AHS human anatomy class, the cellular part of it, is that we challenge the idea of the mother cell dividing to produce two identical daughter cells. This is not true at all, because when the mother cell divides to form two daughter cells, one of those really is a mother cell that maintains the full mitotic capacity and replaces the mother as the reserve cell. So really the mother cell gives birth to a daughter cell that and stays as she was, changed somewhat. You'll never be quite the same, no matter what your doctor tells you after you give birth. And the daughter cell will go on to differentiate into the post-mitotic functional cell. So it's very important to understand that when we think about the adult organism, where our skin cells are dying and falling off, the mucosal cells are dying and falling off, our gastrointestinal lining virtually is replaced within every 4 to 6 days. So we can understand it in that sense. The deep layer of cells are the reserve cells, and they're giving birth to daughter cells that differentiate into the post-mitotic functional cell that lives for just a certain period of time, dies, and goes away. But what we're having with the development of the organism is mitotic division, where there's not a tremendous amount of cell loss. We're just dividing and producing and dividing and producing, and we start with what is called the totipotent cell, a cell that has the potential of becoming anything. So the true stem cell that is totipotent can become anything. Then there are other stem cells along the way that have limited routes of differentiation, so they might be pluripotent. So one progenitor cell that can become all the different types of connective tissue cells, for example, and produce all those lineages, or they might be able to produce all the lineages, the stem cells for producing all the different blood cells, the red blood cell, the different kind of white blood cells. So when we hear the term stem cell out in the public, everybody's thinking that an embryo or a fetus has to die to produce that, but it isn't true. The stem cell is a term that has many, many different definitions, if you will, depending upon how you're looking at the development. So we'll make an example before we come back to this idea of what's going on in the development of the conceptus. Saw an article about a dentist that just happened to collect one of his kids' teeth when it came out, took it right to the lab and put it on ice, took it to the lab, and washed out all the little cells inside it and isolated stem cells from that could differentiate into connective tissue cells, teeth-forming cells, nerve cells, the nerve protective cells, the myelin cells, and things like that. So those are stem cells. But no embryo or no fetus had to die. A kid just had to lose their tooth like they lose all the time. So it's important that as biologists we understand that, because most everybody in the general public, when they hear the word stem cell, all they can think of is abortion. So it's important that we understand that. So we have these cellular lineages of differentiation. The totipotent cell that can become anything. To get that, you either have to de-differentiate a pluripotent cell, which we're not really very good at understanding yet, or you have to collect it or procure it from a very early conceptus. It may not be from the embryo or the fetus. It might have to be from the umbilical cord or something like that. So we need to understand that. So what it means for us is that at any one point in time, when we're following cytogenesis, histogenesis, and organogenesis, we're going to see cells of different potency within a little spot on a histological slide that we might be looking at. They might be multipotent or totipotent or pluripotent, some type of term like that, or bipotent or unipotent. So there's certain levels of commitment as we go along. So this cell and tissue differentiation is a concept when we see that set of terms which should invoke within us this whole picture of this broad spectrum of cells, many of which can be called stem cells, and they're either predetermined to a very limited degree or a not so limited degree as to what they can differentiate into. So this is where we start taking words and putting these concepts and pictures in our mind. Right now we should be seeing one cell, making several cells, making several cells, making several cells, breaking out into lineages that can become a neuron, that can become an adrenal cortical cell, that can become a skeletal muscle cell, that can become an osteoblast, and so forth. So we will see. So now I mentioned the term osteoblast. What does an osteoblast get called once it has become the mature, fully differentiated post-mitotic cell? An osteocyte. So we'll hear this term blast. So a blast cell is a type of a stem cell, but it's typically unipotent, especially in the example that we just gave. So we need this type of thinking in biology because we don't always have to kill an embryo or destroy an embryo. We're not always killing it. We're harvesting cells. We're disassembling an embryo, I guess, in order to get stem cells. Once we understand that full definition of stem cell, then we know there's more places to go and look for cells that are already predetermined. So if we want to produce a particular cell line or a particular tissue, then we go looking at certain locations to do that. So pattern and polarity then become very important. So we've got these undifferentiated clusters of cells that are just kind of moving and dividing and reorganizing. Somehow they're figuring out where they're supposed to go. We'll see that different cells from different layers have got to end up in certain locations in order to produce certain organs. So it's highly organized, and we just look at it in awe in this class. In the developmental biology, they look at all the molecular aspects of what is known about it. So if you really get taken by that aspect of saying, how do they know where to go and when to do this and when to do that, and how is it determined that they differentiate along this line, you may find yourselves taking the developmental biology in addition to this class to open up new avenues of understanding and investigation along these lines. But there's going to be patterns in polarity. We certainly don't want our foot cells ending up at the top of our head and growing out of our head. So something knows or something tells cells to go someplace and develop into our toenail versus the hair that grows on our ears once we turn 30 or 40. Didn't have hair growing on these little parts of my ears, the antitragus, when I was in my 20s, but now all of a sudden I find myself plucking my earlobes, not my earlobes, my antitragus just about every week. So I'm glad it's not my toenails that popped up there some 40 years later. So we have to have the complete symmetry of the organism. If things don't happen the way they should, then we end up with potential aberrations. What's an aberration that happened just recently at Wilford Hall? Conjoined twins. So didn't get the complete destination signal sent to the organs. We go to 250, is that correct? So we're just at this point. So growth and maturation are a key part of it, and then as I just indicated, the senescence of my ears, starting to grow hair, and then ultimately the end point for all organisms, death is a part of life. So we'll pick up with this slide on Wednesday, and we'll push through the rest of Lecture 1 and go straight on to Lecture 2 from there. So I appreciate your time and attention, and we'll see you on Wednesday. Most likely, I'll see you on TV from somewhere else on Wednesday. I didn't even get to use this. Well, you get the website link. You can go and take a look at it. Oh, okay.
I'm going to take a picture tomorrow because I've got
Yeah, that was the ones who work at the health center?
Yes.
Okay.
And I drove past there. Yeah.
Oh, really?
I got to know where at least 4 or 5 people live.
Oh, wow. So where do you live?
I live northeast. I live by Nacogdoches in the Wilson area.
Okay.
It's not so bad. My town I actually tried taking 604 to 37, and that was
Okay. Yeah, what we might end up coordinating, what's your schedule after this class?
I have heart. I have diagnosis of heart failure 2 years ago, and I'm not okay. Yeah.
Oh, okay.
But Tuesdays and Thursdays are better.
That's good. Yeah, the reason I was mentioning it was that